Hello everyone, my name is Rohan Mishra and I work as an assistant professor at the Bioinformatics Center, Savitribai Phule, Pune University, and I welcome you to my lecture series or practical series on protein structure visualization and structure analysis. So I have designed these set of practicals, or you can say I will demonstrate you some interesting facts about proteins, and the objective of designing this set of videos is to orient new users, or you can say orient new biologists or biochemists or even medicinal chemist to understand the structural aspects of biochemistry, okay, or to make you aware of what are protein structures and what wonderful things you can uh, you can learn just by observing the protein structures. Okay, so keeping this in mind, we would be spending a lot of time on analyzing the protein structure, on visualizing the protein structures, and try to gain what exactly do those protein structures say, right? So I'm really sorry. Uh, Before we kind of jump into obtaining those protein structure and perform those surface analysis and perform all the modeling task, well, I think it would be good for you, so that I would kind of orient you on as to where do we get the structural data, or how do we obtain the structural data. Okay, so the objective of this video specifically is to orient you on what exactly the protein structures are, and how do we get the protein structures, how people solve protein structures, and how is that we would judge the quality of protein structures or the models that you obtain from internet. Okay, so to begin with, I usually used to have a general question. Okay, so this photograph is a very famous photograph. Okay, and you might have seen this beautiful picture in any biochemistry, basic biochemistry books. No need to say that this is diffraction pattern of a very famous nucleic acid called as DNA. Okay, this is called as photo 51, and this represent the diffraction pattern that was obtained by Rosalind Franklin back in 1950s. So based on this diffraction pattern, it became possible to deduce the structure of DNA. Okay, so what usually we see in biochemistry textbook is this, and on the very next page you are shown with this beautiful three-dimensional model. So now, just like Captain Jack Sparrow, I used to always wonder as to how do people end up from here, from the diffraction pattern, to the model that has been shown, or to the model that is published. Okay, so this the model of this DNA definitely don't look anything like what is shown in diffraction pattern. So there must be something, some procedure, there must be something that would let people uh, to build model like this, starting from say some diffraction pattern that might look like this. So that is what I used to wonder as to what might have happened from here to here, so that you generate a three-dimensional model. There must be something, and that is usually missing. So early, or you can say the starting students who start out with structural biology don't usually appreciate as to how do we reach from diffraction pattern to your three-dimensional model. and I have created I am delivering this lecture just to fill up this gap. So in this lecture we would try to fill up this gap as to how we end up on the 3D model if we start out with this diffraction pattern. Okay, so we would try to answer this question. Okay, so the thing is that the structure determination of any macromolecule, being nucleic acid like DNA, RNA, or even protein molecules, it don't usually begin with this diffraction pattern. So there are some steps that happen before this diffraction pattern as well. So overall, what my idea of creating this video is to make you aware as to how things start and what is that you usually end up uh, observing, okay, or what is that you end up downloading from PDB, and you might start to visualize or analyze the macromolecular structures. Okay, so to begin with, we would just have a brief preview of the method called as protein crystallography. Okay, so in let us first define, or let us first try to understand what this protein crystallography is. So protein crystallography is a technique, is a biophysical technique, you can say, that is used to determine the structure of a given macromolecule. It can be either protein or it can be nucleic acid or any biological macromolecule. So people or scientific community generally use this protein crystallography for determining the structure of any, or you can say many different biomolecules as such. Okay, so say now you are interested in finding out the structure of a given protein. Okay, or now we can say that you are interested in solving the structure of any given protein. Okay, of any given protein as such. So the first thing that you would need to do is to obtain the crystals for that protein. Okay, and before you obtain the crystals for that protein, it is very important that you learn how to crystallize that protein. Okay, so just a minute. I'm sorry. So the very first step you can consider is that you need to kind of figure out the crystallization condition. And in order to figure out the crystallization condition, there are a few steps that happen before to this step as well. You need to overexpress the protein, and then you need to find out the crystallization conditions, and then you would end up on obtaining the crystals. Okay, so in crystallography, this obtaining the protein crystals is the very rate limiting step, or you can say it is very time consuming steps in crystallography. Why? Because every protein is unique and they would crystallize at very different condition. Okay, so what do I exactly mean when I say that you need to obtain the crystallization condition? Is that you need to figure out some optimal pH or optimal temperature or optimal condition in which the protein will get crystallized. And this is a very difficult task. Being each and every protein being unique, it is very difficult to figure out as such key at what condition, at what optimal condition, at what optimal pH, at what optimal temperature, at what optimal concentration of that protein will eventually get crystallized. So there are techniques like hanging drop techniques by which you can crystallize the protein. So, and as I have mentioned before, this is the rate limiting step in crystallography. So if you heard that it took a researcher say some 10 years to solve the structure of any protein, then you should kind of realize that of that 10 years, that researcher might have spent 8 years in finding out the crystallizing condition for that protein. So this is that time consuming process. So once you obtain the crystal, then the subsequent steps are quite easy. Those are just mechanical and some mathematical formulations that you need to obtain to solve the structures. So the major limiting step in protein crystallography is to find out the optimal crystallization condition, or to find out the crystals of a given protein. Okay, so let me also tell you as to what exactly happens during the process of crystallization. Okay, so we'll say for example, the in the process of crystallization, your objective is to find out ki at what exact physiological condition, at what might be the concentration, what might be the pH, what might be the other factors that might help me to obtain the crystals of my given protein structure. That is what we do in crystallization. Okay, and what exactly, when you figure out as such key, okay, so this is the concentration of my protein that I would require, this is the optimal pH, this is the optimal temperature, these are some other factors that I have figured out. Okay, these are the conditions at which my protein will get crystallized. So what exactly happens when your protein gets crystallized? So we say that your protein molecule, we'll say for example this is your protein molecule, it would kind of orient itself in a periodic fashion. Okay, it would kind of arrange itself in say some periodic fashion, and thus we say that we have obtained the crystal. Okay, so when this crystal will not contain a single protein molecule. Okay, it would contain an array of protein molecules that are periodically arranged in around each other in a specific way. Okay, so your crystal would contain periodically, or you can say symmetrically arranged protein molecule. Okay, and each of this the unit of your crystal can be considered as a symmetric unit. Okay, so your crystal would contain a lot of asymmetric unit. Okay, so now we'll say you have obtained your protein crystals by any of the methods like hanging drop techniques. One more thing, that when I show this image to my student, well, they tend to get an idea that these crystals might be very big, like you might mount it on a ring, but that is not the case. The crystals that you obtain in protein crystallography are very microscopic, and you would need some microscopic techniques even to mount it on to mount it on the X-ray crystallography machine. So these are very small, as it seems that these are present in jars, so that and they might appear bigger. This is just a kind of representation. So so don't get any false ideas as to this would be very big. Okay, so these are very small and microscopic. Okay, so this is what happens in your crystals. The proteins molecule would get arranged one upon another to form a crystal of the given protein. Okay, and once you obtain the crystal, what you do is that you mount that crystal on your X-ray crystallography machine. So if you live in Pune, once you find out crystallization condition, you might take that solution to NCL, maybe, where there is X-ray crystallography machine available, where there is X-ray crystallography facility available, and you would give those crystal to that crystallographer, and he would mount those crystals on your X-ray crystallography machine and expose your crystals to X-rays. Okay, so now you are not exposing a single protein molecule to X-rays. What you are exposing is you are trying to expose multiple protein molecules that are arranged periodically around each other to the crystal, to the X-rays as such. Okay, so now, since uh, there are atoms in your protein molecules, and these atoms will have electron densities around them, and due to those electron densities that are present in your proteins, the X-rays would eventually diffract after exposed to the electron densities from your protein atoms. Okay, so whatever diffracted whatever X-rays that are diffracted, then they are they are exposed to a detector screen, and on due to this diffraction, you would obtain spots on your detector, and these spots will be having different intensities depending on how much is the electron density. Okay, so this is what would happen. If there is nothing, the X-rays would have passed linearly. Okay, but now, due to presence of these electron densities in your protein molecule, the X-rays would diffract, and they would create spots, or these spots are also called as reflections in terms of crystallography. Okay, so what you would end up is the set of reflections on a detector plate. Okay, and you would know their intensities as well. Okay, and your crystallographer would raise up his hand and say, okay, my job is done. Here you have the set of reflection and their intensity values. One more thing, the position of each and every reflection spot on the detector screen is usually represented as H, K, and L. You can say that these are coordinates of, or you can say, using this HKL information, you can say that you can just consider it as these are the coordinates of your reflection on the detector screen. Okay, and moreover, we don't keep this crystal static. Okay, we kind of rotate this crystal when we mount it on that crystallography machine. So the diffraction pattern that you get is kind of three-dimensional, and those the spots that you obtain are usually represented by HKL. Well, this is how you describe each and every spots in terms of their HKL value. Well, okay, so now once you have this, which is the diffraction pattern of your crystal, okay, so now you know that what is the position of each and every reflection or position of each and every spot along with their intensity. Okay, so you would end up with list of position and intensity for each reflection. Okay, so each reflection will have its own position and it will have its own intensity. Okay, so this is data that you have once you have exposed your crystals to X-rays, and the job of crystallographer is over, is finished over here. Okay, so this is what you have observed in most of the biochemistry books. So it is, you know, one of the detector screenshot for as to how many spots are there and what is the intensity of each and every spots. Okay, so now using this data, you need to solve the structure of your protein molecule. Okay, so now what the biophysicist does is that when we have the list of positions and list of intensity of each and every reflection that is obtained from your X-ray crystallography experiment, what he does is that he feeds that value of position and intensity to a mathematical expression called as Fourier transform. Okay, so the objective of you know, giving input of position and intensity to this Fourier transform expression is to obtain electron density map. Okay, so once we end up with position and intensity, our next job is to obtain the electron density for our model or for our protein molecule. Okay, so in order to obtain the electron density, you usually have to use a mathematical transformation called as Fourier transform. That is what the uh, biophysicist would do. Okay, give input these values of position and intensity to Fourier transform and generate this electron density map. Okay, so now let me explain you as to what exactly is this Fourier transform. The general expression of this Fourier transform is shown in this mathematical expression over here, where your this expression rho xyz corresponds to the electron density of atom with position xyz for a spot having the index triple value HKL. So this is how you should read it. Okay, so electron densities are obtained in terms of Cartesian coordinates x, y and z for each and every atoms, or you can say set of atoms in your protein. Okay, so this is something called as expression for this correspond to your electron density. Okay, so in to obtain the electron densities from the uh, from the output of your X-ray crystallography, uh, what things are needed? First, you would require this term called as V, where V is the crystal cell volume. What is the volume of that crystal cell? Okay, and then you see that there is this triple summation. Okay, so this summation, which is now done for each position of x, y and z in a normalized unit cell. Okay, so since there are multiple protein molecules in your crystal, you need to normalize all the electron densities, and in order to normalize all those electron densities, you have done the normalization across the three triple index values of H, K and L. Okay, so from this position and intensities of spots that we know, we usually end up calculating this structure factor, what is called as f of HKL. So this data from X-ray crystallography, from position and intensity, it would enable you to calculate this structure factor. Okay, and in this expression, this I indicate that this is a complex number, the complex number that are usually involved in Fourier series of transform. Okay, uh, okay. So now there is one more factor that is needed to be understood. So using the data that is obtained from X-ray crystallography, you can only infer this structure factor in your Fourier transform expression. Okay, but in order to complete the Fourier transform, you would also require this expression as well, or this quantity is also needed to be measured, and this quantity, what we over here, what we have represented it as alpha HKL, corresponds to something called as the phase. Okay, so you need to know the phase in order to complete your electron density. You would require structure factor, you would require phase information, then you are able to find out the electron density. Okay, so the thing is that you need to separately find out the phases, and this is what it is termed as phase problem in crystallography. Okay, so by using X-ray crystallography, you can only determine the structure factor from this Fourier transform. But in order to complete this expression, you would also require the phase information. And it is said that most of the structural information is hidden in this phase. So you need to perform phasing separately. Okay, so now this phasing is performed, or the phase information is obtained using various techniques like MIR, what we call it as multiple isomorphous replacement. You can separately perform MAD, multi-wavelength anomalous dispersion. Okay, or another easier way to do thing is you can perform molecular replacement if you know that your given protein is similar to some already existing protein. Okay, so well, let us not go in more details of these things. Okay, so it would be enough for you to understand, in order to obtain the electron density map, it is necessary that you should know the structure factor and the phase information. So if you have these two in hand, you can generate the electron density for the given crystal, right, or the given set of atoms as such. Okay, so typically or practically what happens is that the biophysicist would give position and intensity to say some computer program and eventually it would generate the electron density map. This is what happens at the theoretical level in background. So I thought that it would be better for you to know that what is structure factor, what is phases, what is phase problem in crystallography. Okay, so you can define phase problem as unless and until we get the phase information, we are stuck, and just with help of structure factor you cannot complete this expression. So unless and until you have this phase information, your expression of your electron density is calculating electron density is still incomplete. and thus we say that we stuck in phase problem. Okay, so this is how you solve the phase problem, by separately performing experimentation like MIR, MAD, or molecular replacement, and you obtain this phase information and uh, complete your Fourier transform expression and obtain the electron density map. So long story told short, the step that is needed to be done after you obtain these spots, or you can say the position of each and every spot in your from your X-ray crystallography experiment, the objective is to create electron densities. Okay, and that is usually done by implementing this Fourier transform. Okay, so now next thing that is needed to be done is that then this electron density map has got resolution. Okay, you can have electron density map at various resolution. You can say that I have got electron density map at 2.3 resolution. You can say that I have electron density map at 3.0 angstrom resolution, 3 angstrom resolution, 1.2 angstrom resolution, right? So you can get electron density map at different resolution depending on how well your proteins were crystallized. Okay, so next thing that is needed to be done is that what you do is that you would create a model that would fit into your electron density map. Okay, so this is what you do. Once you get electron density map, then you would generate a model that would fit into the electron density map. So what you see over here, well, it is one of the amino acid residue. Okay, this amino acid residue is aromatic in nature. Okay, so you say that if I observe such electron density in my electron density map, then this might be, you can say, any amino acid residue which is having cyclic structure. Okay, and how many amino acid residue contains say aromatic side chain? It can be either tyrosine, it can be either phenylalanine, or it can be tryptophan. Okay, so you can then just guess based on these electron densities as to what amino acid residue might be fitting over here. So if you have such fine resolution of electron density map, you can clearly say that over here you can fit your phenylalanine. Okay, so in this way you try to create a three-dimensional model of the amino acid residue that would fit into your electron density map, and then you would iteratively perform the process of refinement. You would fit the model, create a model that would fit into the electron density map, and then ask yourself a question: have I created a better representation of any model that would fit my electron density map? Okay, then you kind of you kind of ask yourself such question, and you iteratively refine your model by fitting this model into electron density map. And then once you are satisfied as to, okay, I have a model that perfectly fits into my electron density map data, then you would submit that model into public repository like RCSB, PDB, or Protein Data Bank. And it is the model of that protein that you usually download from PDB or Protein Data Bank. It is one of the resource that is responsible for storing and disseminating the structural information. Okay, so now scientific community now can download the model that is deposited in PDB. So what I want to convey you people from this particular talk is that what you download from PDB is some model of any protein, and this model is usually based on electron density map. Okay, so whenever you say that I have downloaded the structure of any protein from PDB, that would eventually mean that you have downloaded the model of that protein from PDB, and this model are built up on, say, some experimental electron density maps. Okay, so this is what it is. So when you download a structure from PDB, you are supposed to say that I have downloaded the model of a particular protein. Okay, and these models are based on, or these models are created on the basis of some experimental on the basis of experimental data in form of electron density maps. Okay, so this is the overall process of crystallography. What you have is this. Okay, and we are going to study the models of various proteins in our subsequent lectures or in our subsequent practicals. So what we would be manipulating, what we would be mostly playing around is the model. Okay, and my sole objective of this video is to make you appreciate that that it is the model that you would be analyzing, and this model is generated based on some experimental data, and the experimental data behind this model are in form of electron density map, which are obtained from X-ray crystals. Okay, so this is how things work in X-ray crystallography. Okay, so now well, let us say that let us talk about how do you judge the quality of a model. Okay, so what I have said is that it is this model that is present in PDB. Okay, so it might completely happen that there might be multiple model of a given protein in PDB. Okay, well, just say for example, if your project guide said, if your PhD mentor said that we need to work on, say, some protein, some therapeutically important protein, maybe cyclooxygenases. You know, the cyclooxygenases are involved in inflammatory response, anti-inflammatory response. Okay, so now you might be interested in finding out the structure, or you can say as to how many structures of these cyclooxygenases enzymes are available in PDB. What is that we know about the structure of this enzyme? Okay, so that structural information is present in your PDB database. So you would then go on, say, any web browser, or you would open up your in your mobile and try to search out as to how many structures are deposited in PDB for these cyclooxygenases. Okay, and when you perform such searches, you might end up identifying that there are, say, some 50 structures that are deposited in PDB just for your cyclooxygenase 2. Okay, now of these 50 structures, how would you determine as to, how would you judge as to which is the best model, so that I can download it and I can proceed it for my subsequent study? Okay, so you need to kind of try to, you should be in position to identify from the whatever structural information that is present in databases like PDB as to what are the best model and which are the model that you are supposed to ignore. Okay, so in order to judge a model, there are you can say practically you can decide the quality of the model based on the resolution and the R value. Okay, so what you have in PDB is this: a model of any protein molecule. Okay, and in order to determine the quality of that model, I said that the quality of a model can be described in terms of resolution. Okay, so when we say that I have downloaded the model with a 3 Armstrong resolution, that would eventually mean that you have downloaded the model that has been created by fitting into the electron density which is having the resolution of 3 Armstrong. The electron density map might look something like this. For this particular model, if it has been created on the basis of electron density map at 3 Armstrong resolution, so this is how the electron density map would look like. Okay, when the author claims that they have a model of any protein molecule of 3 Armstrong, or say for example 2 Armstrong unit resolution, that would mean that they have obtained the electron densities that might look something like this. And at higher resolution of 1.2 Armstrong, the electron density might look like this. Look, the model is the same. Okay, but this model has been created on different realities. Okay, well, say for example, the model that is created on the basis of 3 Armstrong unit resolution electron density map, that means this model has been created by fitting this particular residue at in this electron density map. Okay, and over here you can see that at 3 Armstrong unit resolution, the electron density appears just as a blob of electron densities, right? So you can say you can fit over here tyrosine, you can fit over here phenylalanine, or you can even fit tryptophan. Okay, because this do not allow you to kind of precisely place the atoms in the electron density map. But as the resolution of electron density map increases and you have more fine resolution electron density map over here, you are in no place of confusion as to whether we can fit tyrosine over here, phenylalanine, or tryptophan. Okay, so at 1.2 Armstrong resolution, it becomes very clear that it is the phenylalanine that can fit over here. Okay, if it has been tyrosine, then you might get something like this that would correspond to the side chain oxygen. Okay, so more is the resolution, better is the model. Okay, you can rely on the quality of model. Okay, one more thing that I would like to explain is that more is the value of resolution, you can say the model is not that good. Okay, so you can say that a good resolved structure should have less value of resolution. Okay, so 1.2 Armstrong resolution is better than 3 Armstrong resolution. Okay, so the model that is developed at 1.2 Armstrong resolution is better than the model that is developed at 3 Armstrong unit resolution. Okay, so what you download from PDB is the model, but this is the unique description of the reality based on which those models are created. Okay, so a model at 3 Armstrong unit resolution would be as good as 1.2 Armstrong unit resolution, but it is up to you to decide on what reality this model has been created and appropriately judge the quality of that model. That is what my take home message is. Okay, so the take home message is that when you try to search in PDB and you end up finding out the same protein structure at 3 Armstrong unit resolution, 2 Armstrong unit resolution and 1.2 Armstrong unit resolution, so the best model would be the one which is having 1.2 Armstrong unit resolution. Okay, so that is what the take home message is. So try to work with a model that is having the lowest value for resolution. The lower the value for resolution, it means that the protein has been resolved properly. That is what the idea is. Okay, then this is one way you can judge the structure of you can judge the quality of the model on the basis of resolution. Okay, another way around is that you can judge the quality of model on the basis of R value. So now let me explain what exactly is R value. Okay, so this R value is the measure of quality of atomic model that is obtained from crystallographic data. Okay, so when we solve the structure of a protein, okay, so a researcher first built an atomic model. Okay, and what happens is that I would just explain you. Well, say for example, you have got this electron density map, and this electron density map has been obtained from, say, some diffraction pattern which might look like this. Okay, and on this electron density map you would generate your model. Okay, this is the model that you have generated. This is what happens. This is the this is the diffraction pattern. Then you obtain the electron density map, and then you generate a model that would fit in your electron density map. Okay, so now what the researcher does is that they first built up the model. Okay, and then what he tries is that he tries to create, or he tries to simulate a diffraction pattern that might create, or that might be created if this is the structure. Well, say for example, now computationally what is done is that if you provide this structure, what would be the diffraction pattern? Okay, so from this model you try to computationally predict the what might, if I give this model or this structure, if I give this structure to any X-ray crystallography machine, what diffraction pattern I might get. And then this diffraction pattern is compared to this diffraction pattern, what we call it as experimental diffraction pattern, and this is called as you can say predicted diffraction pattern. Okay, now actually what you do is that you compare the spots of predicted diffraction pattern with that of experimental diffraction pattern, and this R value is a kind of measure how well the simulated or predicted diffraction pattern matches with the experimental diffraction pattern. Okay, so now a perfect fit would have the same spot in predicted diffraction pattern as it is present in experimental diffraction pattern. Okay, so if that is the case, after predicting you obtain same spot at same location as you have obtained in X-ray crystallographic observation, a typical ideal value would be 0. Okay, ideal value should be 0. If we say that the R value for a given protein model is 0, that would mean that after obtaining this simulated diffraction pattern, the position of each and every spot was found to be at the exact same position as in your experimental diffraction pattern. That is what it would mean. Okay, if that is the case, then the R value would be 0. But eventually such ideal conditions, ideal values are usually not obtained. Okay, so the typical values are around 0.20 for R value as such. Okay, so the diffraction, the simulated diffraction pattern would have the R value of 0.20 as compared to the experimental diffraction pattern. Okay, so what people have done further is that they have performed some statistical stuff so as to improve the significance of this R value, and we say that we have this R free value. Okay, so you can compare either R value or either you can compare R free value. Okay, so this R free value, if you find out for a given model that R free value is around 0.26, then the quality of that model is good. Why? Because that is because that is in, say, some accepted range. Okay, so if the R value is around 0.20, then you can say the diffraction pattern matches well with the experimental diffraction pattern. The simulated diffraction pattern matches very well with the experimental diffraction pattern. Okay, so this is what it is. So my take home message is that when you end up searching PDB for a particular model that I am going to demonstrate to you just in a while, you are supposed to look for two terms in each and every structure. Check out what is the resolution and check out what is the R value. That is what I am going to demonstrate to you now. Okay, well, say we, as I have, well, let us stick on to the same example, right? So I want to find out how many structures in how many structures how many structures are there that are deposited in PDB how many structures are there that are deposited in PDB for the enzyme called as cyclooxygenases. So let us check it out. Well, let us go to PDB. You search in Google, go to RCSB homepage. Okay, I think the net is quite slow. Maybe we would just have to wait till it oh, have I bookmarked it? here we go. So uh, follow the RCSB web page over here. Okay, so this is the interface of PhD, and this is interface of your PDB. Okay, this is the place from where you can obtain the structural model, or this is the place where crystallographer would submit their structural data. Okay, now let us search for COX2. You can say I would just simply search for cyclooxygenase 2. Okay, so here you can see that there are a lot of structures for cyclooxygenase. Okay, so well, let us see that cyclooxygenase 1 and cyclooxygenase 2, there are lot of structures, right? So now from over here, well, let us focus only on how many cyclooxygenase structures are from mice. Okay, and you can also refine your search as per the resolution. Okay, well, let us see how many structures are there from mice. Okay, you can see over here that there are some 7635 scientific name of source organism, right? So from mus musculus there are this much structures. Okay, so let us focus on this COX1 for time being. Okay, over here if you click on, you would be on to the taken to the page where you can download the information. Over here you can see that this structure has been resolved at 2.75 Armstrong resolution, and the observed R value is around 0.20. Look, it fits very well. 0.20 or 0.26 is also good. Okay, so R free value is 0.2. What was our criteria? The R free value should be lower than 0.26. Over here the R free value is 0.2. Okay, and you can figure out what is the resolution of this model. So based on these values, R free value and resolution value, you are supposed to determine the quality of this particular model. Okay, and one more thing that I would like to say is that whenever you start to work on any protein structures, when these models are deposited in PDB, these models are published in, say, some paper. Okay, so these models are published. So this particular model, structure of cyclooxygenase in complex with inhibitor celecoxib, which is one of the most important anti-inflammatory agent, well, this particular model was published in this paper. So each model is generated for, say, some specific purpose. Okay, so the author from this paper might have generated this model to see as to, it might happen like, I want to see if I perform mutagenesis in say some protein, what might be its effect. Okay, the objective of creating this model is usually described in such research paper. So when you download the protein for your project from PDB, make sure that you download the paper as well, read that paper as well, and try to understand what exactly that model is. So this is the practical stuff that I want to focus right now. Okay, so just don't kind of blindly download that model and start working on it. Okay, so try to figure out as to where that model has been published. Okay, download that paper, read that paper, try to understand what that model is built for, what was the objective of creating that model, what are the features that are present in that model, and then appropriately start your work. Okay, so the literature that is associated with each and every model, the link of that paper is usually provided over here. Okay, so if you click on this PubMed, you would be carried out, the link will carry you to the PubMed website as such, and from PubMed you can download that paper. So now you can say that this model 3 KK6 was published in a very good journal, proceedings of National Academy of Sciences, PNAS. Okay, so you can download the paper from PNAS, read that paper, and then try to work on your model. Well, that is what I mean to convey from today's lecture. So in our next lecture, we would be talking about PDB flat file format, as to how the structural data is arranged and how do you visualize that structural data. Okay, so this is it for time being, guys. See you people in the next lecture.